So last time we left off talking about photosynthesis and the chemical reactions involved in photosynthesis. Let's talk a little bit about the structure of chloroplast this time, where photosynthesis actually takes place. So, what color are most of the leaves of most plants? That's green. Why are these plants green? Why is most of the stuff that's around us green? Most plants are green. Most plants that are green contain high amounts of chlorophyll pigments. Chlorophyll collects light from the sun and makes the energy available for the reactions of the cell. Where is chlorophyll located in the cell? Chlorophyll is located in special organelles of plants called chloroplasts. Chloroplasts have a very distinct structure that allows them to carry out photosynthesis, and in many ways they're similar to mitochondria, but there are a few significant differences that we're going to talk about. Chloroplasts are triple membrane structures, meaning they have three separate membranes, and you can see that here in the diagram here, and we're going to talk about those three separate membranes. So they have an outer and an inner membrane that's diagrammed here on this figure. Those outer and inner membranes form a double-layered outer envelope around the outside of the organelle. The open space on the inside of the chloroplast is called the stoma. Most of the reactions involved in the actual fixation of carbon, which is the dark reactions, will take place in the stoma. In the stoma, there are stacks of that third membrane, and that's called the thylakoid membrane. So that third membrane is called the thylakoid membrane. You can see the discs that the membrane makes up right here. And stacks of these membranes are called granum. So stacks of these that you see here and the stacks that you see in this GIF here that keep popping up in this GIF, these stacks here, those are called grana. The surface of the thylakoid membrane is going to be the location of the chlorophyll and other pigments, which are going to collect the light and transform the energy into ATP and NADPH. Chlorophyll and other pigments will be organized on the surface of the thylakoid membrane into structures called photosystems that we'll talk about in a little bit. And these photosystems will form the light collecting units within the chloroplast. Now, how does chlorophyll actually collect light? To understand how light energy is collected by plants, we need to talk a little bit about the nature of electromagnetic waves, which can be organized according to the length of the waves, or the wavelengths, as they are otherwise called. Electromagnetic radiation travels in a wave-like manner, so think of this as small particles that move in a wave pattern. Some of the forms of radiation will have very long wavelengths, such as infrared light and radio waves. That'll be stuff down here at this end of the spectrum, while others will have very short wavelengths, like gamma rays, X-rays, and UV light. That'll be down here at the other end of the spectrum. The important thing to know here is the connection between the wavelength of the radiation and the amount of energy that the radiation will contain. So as the wavelength increases, the amount of energy will decrease, and as the wavelength gets smaller, the radiation will carry more energy. So you have more energy being carried down here at this end of the spectrum. As the wavelength increases here, the amount of energy will decrease. The amount of energy in the radiation is going to determine how it interacts with the molecules it encounters. So you'll notice in this graph that the light that we can see, the visible spectrum, which is right here, only represents a small amount of the total spectrum, but this range is still significant nonetheless, because also happens to be the same spectrum of radiation that photosynthesis is active during. So this is also the range which is able to interact with the pigments on the chloroplasts, and therefore it's also referred to as photosynthetically active radiation. That's this word here. So the light that we happen to be able to see, this visible light, also happens to be the same spectrum of light at which photosynthesis will occur. So that's photosynthetically active radiation. So within the range of photosynthetically active radiation, you'll see the same properties as the wavelengths outside of it, And that is, as the wavelength increases, then the amount of energy in the photon, the particle of light, will be less and vice versa. So why is it that only this small region of that radiation spectrum is the only light that can be used for photosynthesis? To understand this, we have to step back and talk a little bit about how electrons are arranged into the shells around an atom. You should remember that electrons are arranged in specific shells around the outside of the nucleus, and the movement of an electron between these shells will result in the energy state of the electron. As you move closer to the nucleus of the atom, as you can see here, the electrons will be at a lower energy state. This means that as you move electrons away from the nucleus, there's an energy that must be added to the electron to allow the movement. When we consider what happens when a photon contacts an electron on the outside of the atom, the energy in the photon will be transferred to the electron, and we refer to this interaction as the photoelectric effect. When the right amount of energy is present in the photon, it'll cause the movement of the electron to the next highest shell around the nucleus. But the amount of energy required is very specific, and if there isn't enough energy, then the electron won't be able to move. Whereas if there's too much energy in the photon, then the electron will be knocked out of orbit around the atom. So there's only a narrow range where there is the correct amount of energy present in the photon. And it just so happens that this correct amount of energy is found within the visible light spectrum. So when the photons of light in the visible spectrum contact the electrons in the pigments, this causes the movement of the electron out to the next electron shell, and the energized electron can now be used for light reactions. If the radiation has a very short wavelength, for instance, if it's in the UV and the X-ray range, then the energy in the photon is too high, and this causes the loss of the electron. If the radiation is in the infrared or the radio wave range, so if it's a long wavelength, then the energy isn't enough to cause a shift in the electron, so the electron on the pigment won't move to the next higher range, meaning that the energy can't be used by the cell. Now, the range of light that can be absorbed by the pigment will be determined by the chemical nature of that pigment. For example, you know that we see leaves as green in the summer because they contain chlorophyll. The color you see is the color that's being reflected by the pigment, and if it's reflected, then it's not being absorbed by the pigment. I'll say it one more time. The color you see is the color that's being reflected by the pigment, and if it's reflected, then it's not being absorbed by the pigment. So if you are seeing green on a leaf, that means that green is being reflected and not absorbed by that pigment. So we can also see this if we think about the ranges of the light being absorbed by the pigment. This graph shows the amount of light absorbed, and the higher peaks mean more light is absorbed. So you'll notice the peaks for chlorophyll A. You don't see a pink and green, because green is the light that it's reflecting, and that's what makes us see green when we look at them. So chlorophyll A, this pigment that we see as green because it is reflecting green, is the primary pigment in plants. There are peaks in both the blue and the red ranges, as you can see here. You see this peak here, and then a peak here. These are the wavelengths of light which are being absorbed by the pigments and actually will be used for the excitation of electrons. That's the photoelectric effect that I just talked about before in the last slide. As we'll see in a little bit, chlorophyll A is the primary pigment and the most important pigment for the plant in using energy from the sunlight. The other pigments in the plant are called accessory pigments. Some examples of accessory pigments are chlorophyll B, which is similar to chlorophyll A, but has a slightly different absorbance spectrum, as you can see here in this graph. There's also carotenoids. One example that you should be familiar with is beta-carotene, which is in carrots. And they'll expand the absorbed spectrum into the green range that you can also see here in this graph. They'll be orange or red in color, and you can see these in leaves that change color in the fall. Another pigment that's pretty common, that's not in this graph here, are xanthophylls. Those are usually yellow in color because they are reflecting yellow light, or light within the yellow wavelength range. The addition of these other pigments in the leaves will help increase the range of the light that's absorbed and increase the energy that's available for photosynthesis. So anything that's not chlorophyll A is considered an accessory pigment that's just going to increase the energy that can be used for photosynthesis. Now let's talk about the structure of chlorophyll. As I mentioned before, and many times, chlorophyll A is the primary pigment in green plants, and this is an important molecule because it will be the only pigment able to capture energy from light and convert it into chemical energy. The other accessory pigments are able to collect light energy, but they have to pass it to chlorophyll A to be able to transfer the energy into a chemical form that the plant can use. Now, how does chlorophyll use energy absorbed from light? The key to chlorophyll being able to absorb light from energy is the ring structure that's in the center of the chlorophyll molecule. This porphyrin ring is able to interact with the photons that come into contact with the molecule, and if they're the correct wavelength and the correct amount of energy, this will cause the excitation of electrons on the molecule. The critical thing to note here is the presence of the double bond in the porphyrin ring, because these double bonds will serve as a means to move the excited electrons within the molecule and prepare the electrons to be used for the next part of the process. In the middle of the ring is a magnesium ion. This is important for stabilizing the ring and allowing the ring to remain stable in the presence of the excited molecule. What would happen if this molecule is linear and not a ring structure at the end? This would reduce the surface area of the molecule and make the collection of photons less efficient, so the ring structure not only helps in the transfer of electrons, but will also be important in the collection of photons. In addition to the ring structure, which serves as the energy collection apparatus, the molecule will also contain a hydrocarbon tail. This region of the molecule serves as the anchoring point of the molecule. That is the end of the molecule that will be covalently bonded to the proteins that attach to the molecule to the thylakoid membrane in the chloroplast. Now, this may seem like a minor detail, but this organization of the molecule means that the porphyrin ring will be placed on the outside of the membrane, meaning that it'll have a better opportunity to interact with the light that contacts the surface of the leaves. So even though it's a lot of detail about the chemical structure of this porphyrin ring, it's important for understanding how it interacts with the light. Now, how is the energy in the electrons used by the cell? As I already mentioned, the ring shape of the chlorophyll molecule will help to increase the surface area that's able to absorb the photons, and this surface area is further increased by the organization of the chlorophyll molecules on the surface of the thylakoid membrane. Now, there are regions which will have high concentration of chlorophylls and other pigments that are arranged in a way that allows the transfer of energy between the pigment molecules. This complex is called the antenna complex, and like an antenna for a TV or a radio, this will serve to collect the energy from the light and move the energy between the pigment molecules and move the energy to the center location of the complex. In the process of transfer through the complex, it's the energy from the light and not the electrons themselves that will be transferred between the pigments. As the energy is transferred, the molecule will return to a low energy state where it'll be ready to receive more light energy. In the center of the antenna complex, there's this special chlorophyll A molecule, which will serve to begin the transformation of the light energy into chemical energy. And it's only here in the reaction center that energy from light will begin the transformation into a chemical form of energy by the process of electron transfer. So you can think of the antenna complex as a funnel which will move the energy to the center of the complex to be placed in the reaction center. So the reaction center, let's talk about that. At the reaction center, there's an arrangement of 3 molecules that will allow the conversion of light energy into the chemical energy. As I said before, in the reaction center there will be a special chlorophyll A, and on either side of this are 2 other molecules, a weak electron donor and a strong electron acceptor. And the movement of energy in the reaction center will take the form of the movement of electrons between these 3 complexes. Before we look at the movement of electrons in the reaction center, there's one thing you should know, and that's the movement of electrons will only move from higher energy to lower energy. It cannot move the opposite direction. The charge of the molecules will prevent the movement of electrons in the wrong direction, and this charge separation will make sure the energized electrons will only move to the next position in the complex. So let's talk a little bit about chemiosmotic production of ATP. And I don't know if I'm pronouncing it right, but that's how I say it. So chemiosmosis is the movement of ions across a semi-permeable membrane down their electron gradient. I'll repeat that again. Chemiosmosis is the movement of ions across a semi-permeable membrane down their electrochemical gradient. This is really similar to what you've seen in the mitochondria with the electron transport chain, and in both cases, the gradient will be a proton gradient produced by the pumping of protons with the movement of the electrons between the photosystems. The protons are pumped into the Thylakoid space, which means that there will be a higher concentration of hydrogen ions inside the space as compared to the hydrogen ion concentration in the stroma, which is where you get that osmosis part of chemiosmosis. And then the concentration difference will drive the movements of protons out of the space and back into the stroma. And the proton movement will be directed through the ATP synthase, which uses the proton movement to form ATP. And this process is essentially the same as the electron transport chain. So let's summarize the light reactions that are involved in photosynthesis. So what you have is water and 2 photons forming O2, ATP, and NADPH. Water is the source of the first electron used in photosynthesis electron transfer. The electrons will be energized twice during the process, in photosystem 2 and then in photosystem 1. As you see here, photosystem 2, then photosystem 1. And then the transfer of electrons will be used to create the proton gradient, which will be moved through the ATP synthase for the formation of ATP, which you see here. Finally, the electrons will be used to reduce NADP, which will provide the reducing power which is used in the dark reactions. So, now let's talk about the dark reactions. These are also called, the main reaction that's at the center of the dark reactions is a Calvin cycle. And we're going to think about the Calvin cycle as a process in 3 steps. So step 1, you have carbon fixation. Step 2, you have reduction of molecules, which will form glucose and generate G3P. And then step 3, you have regeneration of starting molecules forming RUBP. And although the reactions are very different, you should be able to recognize a similarity between the Calvin cycle here and the Krebs cycle, which uses very similar steps in the process. You're not going to need to know all the intermediate steps, but as I talk about them, I want you to note the amount of energy being used in the process and the number of intermediates formed that aren't directly used in the synthesis of glucose. An important note is that the transfer of energy from light to a usable chemical form, in other words glucose, is a very inefficient process, but it's the best method that plants have to make their own chemical energy sources, and obviously it works for them. There's plants all over the place. So let's go through the 3 steps of the Calvin cycle. Step 1, again, is carbon fixation. In most plants, this process is carried out by the enzyme ribulose biphosphate carboxylase oxygenase. or as you may otherwise know it, Rubisco. This enzyme, which is likely the most abundant enzyme on planet Earth, combines carbon dioxide with ribulose 1,5-biphosphate, which is RUBP that I talked about earlier, to form two molecules of 3PG. And in this process, the inorganic form of carbon dioxide, is transferred into an organic form which can be used by the cells for the synthesis of other molecules. Because this process uses a 3-carbon molecule to move the carbon dioxide carbon to glucose, we'll refer to this overall process as C3 photosynthesis. So I want you to know that term because we're going to talk about other kinds of photosynthesis later, but this is going to be referred to as C3 photosynthesis. And later we'll compare this to other forms of carbon fixation in other kinds of plants. So that's carbon fixation, the first step. The second step is the reduction phase, and you can think of this part of the cycle as the reverse of glycolysis. And you can notice here we're using reduced cofactor to oxidize the carbon molecules in glycolysis. So for every 3 carbon dioxide molecules fixed, the reduction reactions will form 6 molecules of G3P, one of which is used for glucose synthesis, and the remaining 5 are going to be used in the final step of the cycle. And this will be a 3-carbon intermediate. And so this means that for every half a molecule of glucose, you'll use 6 NADPH and 6 ATP. So I'll say that again. This means that for half a molecule of glucose, you'll use 6 NADPH and 6 ATP. So that's the reduction phase. And then we have this third phase, the regeneration phase. As I already mentioned, 5 of the 3 PG molecules formed in the reduction phase will be used in the regeneration of the starting molecule, which is RUBP. And this process takes several steps that aren't shown here in this figure and are going to require 3 additional ATP molecules. So in summary of this whole process of the Calvin cycle, in the synthesis of a single glucose molecule, you will use 18 APT. That's a heck of a lot of energy to just get one single glucose molecule. So like I said, it ain't the most efficient way to get energy, but there's a lot of plants, so it works for them. So let's do a little comparison between chloroplasts and mitochondria because they have really similar processes, and I think it's a really great way and so does Brother Albert because I'm learning how to teach from him. It's a really great way to understand what's going on with chloroplasts and how it's making energy. So in plants, you have both chloroplasts and mitochondria, but when the sunlight is low or in parts of the plant where there's no chlorophyll, such as the roots, the glucose, which is formed in the leaves, will be used to supply ATP for other purposes in the cell. So both are going to be active, both the chloroplast and the mitochondria are going to be active in plants at the same time, but you should be able to see the similarities between the processes in both organelles. Both will contain electron transport chains, but the source of the energized electrons is going to be different for them. In chloroplasts, water will be split to provide the electrons, which will then be energized using light energy. And then in mitochondria, NADH and FADH2 will be the source of higher energy electrons, which will be formed in the process of the Krebs cycle. And so in other words, in the oxidation of glucose molecules, as they pass the electrons through the proteins, there needs to be a terminal acceptor in this process. And in chloroplasts, NADPH will be the terminal acceptor, forming NADPH. And in mitochondria, oxygen is the terminal acceptor, which will form water, making a reduced form of O2. So those are some differences between them. You should also be able to see that the two processes in many ways are working kind of in the opposite direction. Both chloroplasts and mitochondria will use the transfer of electrons both chloroplasts and mitochondria will use the transfer of electrons through proteins to pump protons. Too many P's. And the established proton gradient is going to be used with ATP synthase and the synthesis of ATP, which will be used in the cell for other purposes. So you got the same type of mechanism going on with the exactly opposite outcome. In the chloroplasts, energy will be used to fix carbon dioxide to an organic molecule, and in the mitochondria will oxidize the organic molecule in the formation of carbon dioxide and ATP. And next time, in the next video, that's when we'll talk about photorespiration. So that's it for this lecture video. That was a lot of chemistry to digest. I recommend that you watch this video again, and then watch it again for a third time, because there's a lot of chemistry in there, and I know a lot of you are biology majors. So make sure you spend a lot of time with this video, and if you have any questions, go ahead and post those on the discussion board on Schoology. Okay, see you in the next one. Bye.